Hello everyone. Today we are discussing about bacteriophages. Phages, formerly known as bacteriophages, are viruses that solely kill and selectively target bacteria. They are most common biological entities in nature and have been shown to effectively fight and destroy multi-drugs resistance. ബാക്ടീരിയോഫേജസ് എന്ന് പറഞ്ഞാൽ വൈറസസ് ആണ്. അത് ബാക്ടീരിയസിനെ ടാർഗറ്റ് ചെയ്ത് കില്ലുന്ന വൈറസസിനെ ബാക്ടീരിയോഫേജസ് എന്നാണ് പറയുക. Phages. Bacteriophages are viruses that infect bacterial cell after injecting their genetic material, DNA or RNA, and kill them. Two bacteriophages are there, lambda and M13, which are used as cloning vectors. The genome contains an original replication gene for head and tail protein, an enzyme for DNA replication, lysis and lysogeny, and single-stranded protruding coaxial end of 12. ബാക്ടീരിയോഫേജസിൻ്റെ ജീനോമിൻ്റെ അകത്ത് 48,502 ബേസ് പേഴ്സ് ഉണ്ട്. One of the many bacteriophages lambda cloning vectors that have been devised as two BAMH1 sites that flank the integration or accession region. When purified DNA from this bacteriophage cut with BAMH1, three segments are created. The left arm contains genetic information for production of head and tail, and the right arm carries the genes for DNA replication and cell lysis. And the third one, middle fragment, that has the gene for integration accession process. The objective of the genetic engineering protocol is to replace the middle segment of lambda DNA with cloned DNA that is approximately 20 kilobase pairs in length. The source DNA is cut with either BH1 or SO3A1 and DNA pieces that are 50 to 20 kb are selected. Two samples are contained and incubated with TA4 DNA ligase. Then empty bacteriophage head and tail assemblies are added. Under this condition, 50 kb units of DNA are packaged into heads and ineffective bacteriophage particles are formed. Once inside the cells, the recombinant viral DNA is replicated. There are two types of vectors. First one is replacement vectors and second one is insertion vectors. Replacement vectors. These vectors have two restriction sites that flanks a region known as buffer fragment. This region can be replaced during cloning into this site. Example, EMBL4. The last one is insertion vectors. Vectors has single restriction sites. Replacement vectors അകത്ത് രണ്ട് restriction sites ഉണ്ട്. Insertion vectors അകത്ത് ഒരു restriction sites മാത്രമേ ഉള്ളൂ. Example ആണ് lambda 10 and caron 16A. The phages is cleaved with a restriction enzyme that cuts it only once and target is inserted into this site. No phage DNA is removed, therefore a much lower size of DNA can be inserted. Lytic cycle of bacteriophage, which also commonly referred to as reproductive cycle of bacteriophage. There is 6 stages in lytic stages. The 6 stages are attachment, penetration, transcription, biosynthesis, maturation and lysis. Lysogenetic phage എന്ന് പറഞ്ഞാൽ lysogenetic phages incorporate their nucleic acid into the chromosome of host cell and replicate with its unit without destroying the cell. Thank you.